approach where UniRef really focuses on just clustering our data out there into some arbitrary cutoff, right? So instead of trying to figure out this is a gene that does this annotation, let's forget the annotation side of it and just literally cluster all sequences at either 100% identity, which means they're identical, or 90% identical, that'd be UniRef 90. So everything within that cluster would be 90% similar or higher, or UniRef 50. That's really nice. It's comprehensive, right? Because you can imagine you can easily scale that up over time, keep a catalog. The downside is that they would not have a lot of annotations for those clusters by themselves, but you can, map those clusters to annotations afterwards. And so UniRef will actually come up a couple times. We're going to use it in our lab, and it's also used by the Human 3 pipeline that I'll talk about as well. If we talk a bit more about function, I think Keg is still really well known out there, and we'll talk about how that breaks down. It's been around for a long time. It went private. Oh, I don't know how long it's been now. 6, 7 years, maybe? I don't know. It's been quite a while. So you do have to pay a license fee for like newer versions of it. But even on the previous versions, which was made public, people still use the Keg quite a bit. Full access does require a licensing fee now. And then I would say very tied with Keg is Metapsych. And so Metapsych is fully free pretty much as far as I understand. It's very popular as well. It's probably seen as a major alternative or replacement for the Keg. The Keg comes out of Japan. Metapsych is out of the US. But Metapsych and Keg have very common sort of structures in that they map ortholog groups to larger modules or pathways and really focus on model organisms and expanding those to other organisms. So these are the slides I had about Keg, and I was thinking about trying to update it to Metapsych, but I didn't, and I think that's okay. They're very similar structure, and I just want to walk through sort of how the Kegs organize these things. And a lot of these things will also port over to Metapsych. So Keg groups their sort of smallest fine-grained functions into what are called Keg orthologs. They're the most specific, and they're thought to be essentially homologs doing the exact same functions, or orthologs. There, at the time that I wrote this slide, there was about 12,000 KOs in the database. So if you had a license, that's expanded dramatically. And then those Keg orthologs can be grouped into Keg modules and Keg pathways. And I'll talk about that in a second. They have identifiers like you see there. You'll often see them in manuscripts, K01A03, et cetera. And this is just an example for K01A3 that has, if you go to the website, it has a name for that, a definition, shows you what pathways and modules it's involved in. And sometimes there's even information about human diseases. If it's a human ortholog in human, they'll link it to disease. So if we scale that up to Keg modules, essentially these are groups of Keg orthologs that are defined in some modular functional unit. So not quite at the scale of a pathway, they're a bit smaller than that. At the time of this, there was 750 Keg modules, and they have this identifier starting with an M. They're a bit interesting in that, I know this is really small, unfortunately, but they have this unique structure in that they're not just a bag of genes altogether. They have really defined things that complete this module so that they have a common function. So in order for this particular module, M0002, you essentially have to have this very first gene, this very first Keg ortholog, and then at this next step in that module, you can have different options. So you could just have this one ortholog, or you could have one of these two and this one to go to the next step. At this next step, you just have to have one of these 4 Keg orthologs. Finally, you have to have this Keg ortholog and then one of these 2. And so it defines that structure, and to say, hey, this module is present in this organism or in this community, you would actually have to, validate that structure. The next one up is pathways, and so that actually doesn't have that structure. It's still joined together, and so this is a nice pathway shown on the right with it highlighted in pink. This is also what you would see traditionally in a lot of manuscripts where they link, Keg orthologs into pathways. And they have these nice graphical maps I'm sure you've seen in different papers where they'll highlight different steps in metabolism. You can then eventually collapse those pathways into even higher level things like amino acid metabolism or carbohydrate metabolism. At these points, you would have, the other thing I should have mentioned maybe is EC. So those are enzyme classification. And so we'll see that those IDs are used both in Metapsych and also in Keg. And so they're embedded in the system and are used to eventually map. The sort of take home here is that what you'll see is that there's an initial annotation stage where we'll say this gene is this UNIREF or this Keg ortholog or this EC number. And once you have that, you can then usually convert it by mapping those annotations up to modules or pathways or even to other systems fairly reliably just by mapping them because we have that in databases. But you have to get that initial classification to say Keg ortholog, or what we'll see often is like an EC number or a UNIREF ID. Okay, so that's just a little overview of annotation databases. I guess, is there any other databases that I didn't talk about that I should have, that people know about? Okay. So if we start to think about annotation systems, like systems to apply now these annotations, there's a few different that we can look at. There are web-based systems out there. So if we look at things like MGRAST, I mentioned earlier, as a website where you would literally load up your data to the website. IMGM, which is the Integrated Microbial Genome Database, and the M stands for Metagenomics, is out of JGI. So they are essentially sort of like a repository for both metagenomes and genomes, but you can also upload your data to their server and they'll do annotation. And the same thing with what used to be called the EBI Metagenomics server, which I preferred because it was obvious where it was at EBI, called it Magnify. Again, upload your data. You're going to wait for some time. And it's going to spit out results at the other end, which you can sometimes look at your results in context with their other metagenomes. Pretty good systems. Downside is probably that you just have to wait. And then once you sort of do that, you're sort of stuck in their system. There's not, you can download tables, I should say that, and do other things, but you're sort of a little bit stuck in their framework, I guess. So it's just lack of custom work, I guess. Should always mention, I guess, what's been around forever is Megan. So this is a graphical interface tool. So instead of command line, you can download Megan on your computer. Usually requires some sort of similarity searching to be done in the past through BLAST or Diamond. You then feed those results into Megan. And then Megan does the assignment for taxonomy and function. And then you get to actually browse that data in a graphical interface. So very satisfying. Still gets updates as far as I know. A pretty good system on its own. And then what I'll call local based or sort of custom pipelines, which you would operate from a command line. There's many out there with metagenomics. So compared to 16S, we have Chime and Mother, and then there's a few plugins and offshoots. Metagenomics starts to go absolutely crazy in the different ways that you can do taxonomy, and then function, it gets even crazier. Mostly because, people have different preferences, and later on we'll get about, what you're actually going after will dictate maybe how you do your functional annotation. But definitely from like a first pass or a major use, there's a few out here that I'll mention. So Carnelian is a K-mer-based approach. I put it here because it actually just doesn't take raw reads. It actually does gene calling ahead of time and then maps those out. Human 3 is probably the most popular method out there, and you've probably heard about it before. It comes from Curtis Huttenhauer's group, and I'm going to talk about it in a little bit more detail so you understand how it works. Or there's custom pipelines out there, and so for this example, we'll be using one from Microbiome Helper, where we sort of come up with our own custom pipeline that uses common similarity search tools against common databases like UNIREF, and we've put those together into a tool we have not called anything yet, I guess. I don't know if it's worthy of calling something, but I'll just call it a custom functional annotation pipeline. So what are the challenges in functional annotation? So some of the same challenges we had with taxonomy assignment are obviously in functional annotation. So partial gene fragments, right? If we're trying to annotate reads, those reads may only partially cover a gene, most likely, right? We're talking about 150 base pair sequence. A common gene length is around, it's a common gene length. I guess it'd be 1,000 base pairs, right? Or 1,000 base pairs. So best case scenario, you get all the gene, you're still only going to get 150 base pairs of, say, an odd average 1,000 base pair gene. Not great, right, for identity. And sometimes, obviously, it's not all coding, so you could have just a fragment of the gene. You might only have the start of it, or you could have the end, and so that's challenging. We have mixed communities again, right? So we're talking about functionally annotating things. Viruses obviously have a huge repertoire of possible genes out there. Most of them we don't have any idea of their function, and so those out there as well, and in eukaryotes as well. And so we have to be cognizant of that when we're annotating something. It might not always be bacterial. We could be annotating it from whatever organism it's from a functional level. And again, of course, computational bottlenecks are real here. So speed is still very much important. If we thought taxonomy assignment was slow, functional annotation is going to be slower and takes time. So what's a bit unique, I guess, to functional annotation a little bit more here is that there's potential biases from different things. So one is gene lengths. So if we're talking about counting up the presence of a particular gene, right, the chance that we would sequence a larger gene is higher than sequencing a smaller gene, just because at random we're sampling. And so you can imagine if a gene is 1,500 base pairs and another one is half that, 750 base pairs, we're twice as likely to sequence and then twice as likely to get double the count of this gene, even though, you know, they're both present at one copy. Does that make sense? This bias of gene? Yeah. And then the other big thing is inferring modules and pathways with multiple organisms in a mixed community is definitely not straightforward. So I sort of watch how that would work in a genome, right? How we map KOs to modules and the pathways. How we do that in a mixed community is a lot more complex, and we'll talk about that in a bit more detail. Okay, so the key steps of a functional annotation pipeline. So first, similarity search are our big go-to. So mappers like BWA and Bowtie are very fast, but they are limited to DNA space. That's okay, your query read is DNA. But a lot of these functional annotation databases are proteins. They're protein databases. And so that means that it's hard for us to search. We can search DNA versus protein, but we have to use different tools like Diamond or MMSeqs that allow us to do that searching in protein space. So we can search in DNA space, but it's not as sensitive. It is fast as Bowtie and BWA, but we're limited in what we can do there. So then for protein alignments, there's tools like Diamond, MMSeqs2. MMSeqs2, I guess, is faster than Diamond, is sort of the new kid on the block. They work in protein space. They're fairly sensitive. They're what would be comparable to something like a BLAST-P or a translated BLAST, but they're way faster, like tremendously faster. I think Diamond was around 1,000-fold faster than BLAST, and I think MMSeqs is maybe, I actually don't know the number, but significantly faster than Diamond. So we'll use these tools to do that similarity searching. Of course, always at the cost of that is probably some accuracy loss, but we're talking about just mapping reads to genes. We don't really care if it's 75% or 70%, or maybe that will miss some lower homology maps. We're usually looking for fairly high identity maps. We're looking at things like 80% or greater and mapping those and just taking the top hit. We don't really care about the other hits. And so that works good for these tools. And then for database, obviously that comes into play a lot about what is our database choice. As I mentioned, we'll be using Uniref, but depending on what you're interested in, we could use smaller databases that are targeted on functions that you're interested in. So we'll talk a little bit about those, but things like virulence factors or something, or antimicrobial resistance genes, there's particular databases that are specifically focused on those. And so your searching then might be focused on those versus, you know, UNIREF 100, which is huge, right? So after that, you know, after that mapping phase comes this, a few steps over normalization. I put normalization in quotes because it's probably the most overused word in bioinformatics, which means probably about 20 different things to 20 different people. But here what I'm trying to say is that we correct for this gene length is probably the most used normalization method. So what we do, and this happens with transcriptomics and humans and things, we scale our counts by the length of the gene, right? So we know the length of the gene, and we essentially just divide our hit number to that gene by the length of the gene. So that means those larger genes will get scaled appropriately downwards because we're more likely to sequence them. The hope there is that the numbers that come out equal, you know, counts of the actual genes, units of genes. So because of that, you'll often hear this term that results are reported as RPKM, which is reads per kilobase per million. And so this is a carryover again from transcriptomics, where we'll do this a lot. So things are scaled by the gene length, and then they're reported as reads per kilobase per million. On top of that, there's other methods out there that will do some other scaling. So you can imagine if you have a hit of a sequence to a database, there might be a bit of a difference between in your head, well, this maps, you know, at 80% identity to it, or it maps 100% identity to it. Should we scale that based on that identity? If a hit is a hit, does it really matter? Probably not. Some people will try to think about scaling your numbers based on that. Some will also take into account genome size, right? And factor that in. And then also sometimes there's just other scaling factors to make the numbers more usable, right? So instead of dealing with really, really small decimals or something, they'll multiply by a common factor across the board to essentially make them look like real counts. So there's lots of weird little things in there, but essentially you have, you know, you have your hit counts, you divide usually by gene length, and then you might do some other tweaking to spit out numbers that mean slightly different things. But they're all getting at this idea of, we see this many genes or gene-like things in our sample. So pathway inference is super complicated, and I just want to present it here so you appreciate why it's complicated, not go into great detail about, you know, this a lot. And I know this is sort of text heavy, so I'm sorry. But the idea here is we're mapping our genes to those, say, those Keg pathways. And the result is that we could have many spurious pathways. And that is basically, if we just read this second part, is that, if we read this point right here, say if a pathway has about 20 genes, and we find 2 genes in our environment, in a particular sample. So do we count that pathway, like do we count that as 1? Do we say that that pathway even exists if it's only 2 out of the 20 pathways? You're like, well, not maybe. What about 10? Do we call it that it exists there, or do we just say it's a half a pathway, or what? Or like how do we do that? And so the reality is yeah, sure. If we have 18 out of 20, that sounds great. We'll count that pathway as existing. But maybe those same genes are in multiple pathways, and so those 2 genes out of 20 are actually like 2 in another one that only has 5, and it really belongs to this other pathway. So there's tools out there to try to essentially remove spurious results and I would call smoothing out our pathway inference. So MinPath does this. MinPath was created for genomes to actually like remove these spurious pathways and link what are the true pathways present in a genome. But it was meant for genomes. And so you can imagine that this gets really more complicated now if we're talking about a mixed community, where not only do we not even know if it's in the same organism, it could be multiple organisms. So how do you know if these 3 genes out of one pathway are even in the same organism? Do we count that that pathway exists? Depends how you think about it, right? I mean, maybe if this organism is handing off products to another organism, then that pathway exists, right? But it might not. And so we don't have information about handoffs, points, and it gets pretty complicated about how do we do that reliably. I don't think I want to go further than that. But it makes that step usually an extra step, and it's not as simple just mapping, oh, so we got this KO, we have this pathway. There's usually some intelligent things going on under the hood to try to improve upon that process. The other thing just last about this is that we don't always have the sequencing depth. So maybe those other genes are really there. Maybe those 18 other genes are there, but we just didn't actually sequence deep enough to find them. So when do you start to say, well, those are probably there, we'll start doing some smoothing. Okay, so it's a complicated problem. A few people have approached it in different ways, and the tool that has handled this one, and I'll talk about it a little bit more here, is Human. So I previously mentioned Human. It's widely used, especially in human microbiome applications. It comes from Curtis Huttenhauer's group, and it's a really nice pipeline, to be honest. And so it is on version 3 at this point. And I'm just going to go over the sort of tiered search, because I think that's the fundamental units of it. And at the end, it does do this pathway smoothing and other things under the hood as well. But what makes Human a little bit maybe unique, or a bit, I think a bit intelligent a little bit, is it does a couple of things to make things faster in I would say a pretty intelligent way. So this graphic is actually from Curtis's group, so I've borrowed that, and I've just broken down the major steps. So the idea here is you have your input sequences, and what happens is you use, the first thing you do is you actually do taxonomic assignment. So under the hood, Human 3 runs Metaflan 3 to say these species are present in your sample. Based on those species being present, it then makes essentially a custom database of those species to search against. And it does that search with bowtie, right? Because I remember I said bowtie is really fast, but it's only nucleotide versus nucleotide. So it says these species are here. In this example, that would be this light blue and orangey species. And now it's going to map reads to those genes in that species, or pangenomes actually. They go a little bit further and they make pangenomes, collections of genomes around that species. So that's cool. So that's essentially what they call this taxonomic pre-screen, because it's fast. Your database now is only the species in it, which is much smaller than all organisms in the world. And the hope there is that you remove a lot of your reads. You basically filter those out. Then afterwards, they do their second tier here. So this is, that's pangenome mapping. And then their third tier is then what I would say most other custom tools do, including the one we use today, is they just take any reads that are left over and they try to similarity search those against a protein database using a translated search. In particular, they use Diamond, and then they do that Diamond search of your any leftover reads against that. So the idea there is it speeds things up. Does that make sense? Fast search, remove hopefully a lot of it, and then you'll map a lot more with the translated search. And then even the reads that are left over, they'll spit out if you want them at the end, saying, well, we don't know what to do with these reads. They didn't map to any proteins. And the database they use by default is Uniref 90, I believe. It's Uniref for sure. I think it's Uniref 90 by default, although there is some selection in there between Uniref 90 and 50. The other nice thing about this is that this mapping to those species gets us into this idea about stratified output. And so I've been on this train a little bit about how we can break down the functions that we identify by the species contributing to those functions. So this is just a slide showing, okay, we have found this particular Uniref 90. So you understand now what a Uniref 90 ID is. Has this other gobbledygook ID. They have a little description of what this gene group is, an IMP dehydrogenase. I have no idea what that is, actually. It's pretty sad to me. And then they have a count. So it's 600. So this is their scaled count. And you can get this result just by itself. So that would be if you ask for unstratified results, and this goes for Human 3 as well, you would just get this first line, and then you would get another line with your other Uniref group. But if you ask for stratified output, you then can get this information about what species, or unclassified, contribute to that same function. And so that happens through their mapping to species with metaflen. Does that make sense? This idea of stratified? Okay. So this is the colored version of that, which I should have shown first. And then this also shows you then that you can get things like pathway information. They'll report sometimes coverage in a sample. I don't find that very useful, where it's literally just binary. One, we think this is in the samples covered or not. Or they'll give you pathway abundance, which again is this sort of gap-filled magical min pathway with a lot of parameters, figuring out what the count is for this pathway. So that's Human's approach. And that's the basics, I guess, of how it works. So with this breakdown of functional contributions, we can do some sort of interesting, what I think is interesting. So if we just talk about a particular function and we just focus on this top figure A, right? We could look at a particular, let's call this a particular pathway, okay? And we could look at its variance across our samples. That's all this would show. This is just these gray bars. But under the hood, there could be a lot of information about, well, what species are contributing to that pathway under the hood? What does that look like? And is that interesting? And so these are just 4 different sort of versions of what that breakdown might look like. So this was described as contributional diversity within Huttenhauer's group. And so we can use that term. And this is just an example of a review paper that a PhD student and I wrote. So we can have some, this is just showing like a high diversity sort of situation where multiple species are contributing to a particular function, but it's fairly even across the samples, right? This is fairly high diversity, but different organisms are contributing to that signal. This is sort of low diversity. So basically one species contributing all that function, and that's pretty even. And then this is cases where we have, that function contributed by usually a single species or a couple species, and it varies. And each of those tells us, possibly different things, right? It gives us a bit more inference about whether this function might be of interest. Things like this, where you see this really steady, could just suggest we're looking at this core function that's staying about the same. Things like this, where there's a lot of different species contributing to that function across communities and it varies, could suggest that maybe that function is important at the community level. And maybe it leads to other things that we could talk about. So this has been a bit of a session with Fermine for a while, and we've applied it in a couple of papers, which I don't have examples of today. But we've also been working a bit on a visualizer to also handle this stratified functional information. And so this was recently came up with the name Jarvis by a member of my lab. So it stands for just another stratified RPKM visualizer. I guess Dewani is a Iron Man fan. And so that's what we called it. And the idea with this, and you get to play a little bit with it later, it's definitely like early on and we want to improve a lot of it. But the idea is to not just show, function and taxa separately, but really try to get those groups. And so this is data that you'll basically generate today between HMP data from Crohn's disease and non. And then we can map essentially those samples to taxa, and then also then to the pathways on the right. And we can visualize, how those contributions to go together. So in this middle one, we have a particular family. You can see how that's connected to samples in CD versus non, and then also to this particular pathway. Definitely, there's lots of things we want to add to it to make it more usable, add in some statistics, a little bit better mapping through. Right now, it's a bit static, but it's our first attempt. And so be nice, but I'll take criticism on it as well. That'd be great. Okay, so if you've been sitting here through all this and you're like, well, I only got 16S data, I'll return us to this slide a little bit in that, of course, we can also use things to infer function from 16S data. And I used to talk about PyCrust a lot more detail, and I just wanted to briefly mention it here because we can use things like Jarvis or stratified output directly from tools like PyCrust to also get functional information from 16S data. So at this point, if you're like, I don't know what the hell he's talking about with PyCrust, I will explain briefly what that is. So at this point, we've had data where obviously yesterday you spent a lot of time with 16S data, eventually getting to a table where you have ASVs or OTUs by your samples. And in that table is obviously the basics of calculating things like alpha diversity, beta diversity, which would make PCA plots, heat maps, box plots, just beautiful things. And obviously now metagenomics, this morning we covered about how we can take that metagenomics and also get taxonomy tables, right? It would be very similar to this, hopefully. We know it's not going to be the same, but we can also do really similar things with that from a visualization standpoint and a statistics standpoint. And then we just talked about how we can then, take that metagenomics data to get functional information. So it's a similar table, right? But now we have Keg orthologs or pathways also by samples. And again, we can do similar things now in sort of functional world. And then PyCrust was this tool I previously mentioned, essentially, which takes, your 16S data and these abundances to try to predict what the functional information would look like. Now, I'm not going to go into great detail about this. Essentially, it uses obviously reference genome databases and a phylogeny to essentially try to predict what those functions would look like. But the result is that you would get from your 16S data, EC numbers and metasite Keg pathways. You can also get things like IMG phenotypes, which are just broad functional categories. Pathways are inferred using the exact same pipeline actually as Human for using MinPath to smooth that over. And as I just mentioned, because it's predicted from the 16S data, inherently it has that stratified output in it. And so you can put that into things like Jarvis as well. Now, PyCrust has been used heavily, and I usually go on a bit about this more, but obviously it has its limitations, and so I'll mention that right up front. It's not good in all cases, and so it really does just predict community gene abundances. I'm not naive to that. And so because of that, there's lots of caveats. Obviously, with all these things, not just this, but metagenomics, we're not talking about active molecules, right? We're not talking about proteins and metabolites. We're talking about genes. And accuracy could be very low on individual genomes. So PyCrust does fairly well because it ends up getting accuracy across a community of organisms, which helps its accuracy overall, but it could do really crappy, on individual genomes. And also what's probably not appreciated very well is that the accuracy is high. I would almost say inflated. That sounds maybe detrimental, because inherently it's studying enzymes that are well studied. So if you think about E. coli is our classic case, right? We sequence E. coli genomes, and it's like, well, how, E. coli genomes are 100% identical at the 16S level. And we know that there can be vast differences in the gene repertoire of different E. coli strains, right? And so, well, how could PyCrust predict that? And it can't. That's the reality, it can't. Now, the thing that I'm trying to get in favor of it here, what helps it, is that a lot of those differences in genes are actually in genes of unknown function. So the things that are more variable tend to be not annotated well. And the things that are conserved across genomes, those are conserved fairly well. And so because we're annotating them, we do pretty good on the things we know about. The things we don't know very much about, imagine AMR genes, phage, prophage, things like that, we would not do so well. So anyway, that's my just disclaimer. Obviously, you can use it. Just watch your language around it. And obviously, it's for hypothesis generating, and validation is always a very good idea. Okay, so this is sort of the standard pipeline, right, of how we go through everything. Just coming back to annotation, I wanted to sort of mention a few of these categories about specific databases or specific types of gene annotations you might be going for beyond, the standard UniRef pipeline within Human. And so that depends on your research questions and other specialized tools. So I mentioned earlier, virulence factor database. If you're really interested in virulence factors, if you're looking at pathogens, there's things like the VFTB, the virulence factor database, or the CARD database for mapping essentially your genome, your metagenome data to those databases. And so that might not be captured, by just simply running it through Human. Antimicrobial resistance is very related, obviously, to the CARD and to the virulence factors, and is obviously a subset of that. This is not my area of expertise, but Rob Biko, who you heard about from yesterday, has spent a lot of time on this with Fiona Brinkman, and they've developed a tool called Arete, A-R-E-T-E. So if you're interested in that, you can Google it. They have a GitHub page. I would say that they're at the forefront of that. There are other methods out there as well for especially mapping metagenomic data to AMR. And so I don't mention them here, but I feel free to ask Rob Biko maybe on Slack or send them an email. I'm sure they would love other people testing out their code. Some people, specifically in human gut microbiome, sometimes are really interested in different types of carbohydrate metabolizing genes. And KZ, C-A-Z-Z-Y, has been used in the past. I haven't seen it maybe as much in recent years, but it's very popular for a while to map things to different types of carbohydrate metabolizing genes. And it gets at a very precise level that's not captured by something like a UNREF database. And poor phage, they never get much of attention. And this is all I'm going to talk about. If you are interested in viruses, it's going to open up a whole other kettle of fish. And so there are specific methods if you're seeking out not just function, but really taxonomy. You're trying to pull out viral sequences from your metagenome. I mean, I should really spend more time on this. It deserves it. But there are lots of tool development out there for pulling those reads out, trying to annotate them from a taxonomy assignment and functional assignment. Some of those are aimed at obviously free-floating phage, but also at prophage as well. So within there, VirSorter and VirFinder have been around for a while, as well as Marvel. I quickly looked yesterday, and Vibrant, I think, is the new kid on the block there, so they're the best. I have no idea which one works the best, but you know, they do things like looking at viral databases, obviously, to try to pull those out. They also do some really neat things that are sort of unique to viral data, in that, you know, viruses really put their genes close together. They really jam them together, right? So if you do assembly first, you can use that coding density to predict that that's a viral or phage gene or genome, right, based on that information. And they use even some other composition-based approaches to improve that. It's a whole other area by itself. If you're interested, I can talk about it separately. I've dabbled in it a bit, but I'm definitely not an expert in it. And then I also want to mention that there's sometimes these large, what I call microbiome catalogs. So when I talk about UNREF and things like that, those are usually based off of reference genomes, and they've clustered genes within these reference genomes. You can imagine now, with all of the MGS sequencing that's gone on, that we have a lot of data that's not captured by reference genomes. And specifically, you know, if you think about the human gut, we have generated a lot of metagenomics in the human gut. So why don't we, you know, zone in and focus on clustering genes within the human gut? And that's been done, especially by, is that Pierre Bork's group? Does anybody know? They had like a gut catalog. John, do you remember this gut catalog? I'm putting you on the spot all of a sudden. Okay, anyway, it's been done by a European group. It's not very descriptive. I do have an example of one that includes human, but also environment. And this is kind of a nice big paper that came out last year. They have a nice online website where you can download or map your sequences to their large gene catalog. And so these cover lots of different habitats. And the idea is, you know, if you want to try to maximize the number of reads you can map to something, to some unit, well, then you should try to get the biggest database you can. So this is what they're going after. Okay, and I think I already did this warning that essentially with all of this, we're not looking at, you know, transcripts or proteins. We're looking at genes. So that doesn't mean that they're active. We have no idea. This is a nice segue to John for tomorrow, right, where he's going to get into metatranscriptomics and how that provides a lot more information to layer on top of just knowing that a gene exists within our community. And as I previously mentioned, this DNA might be even from dead cells, right? So this could be just DNA that's degraded, especially if you're talking about human gut. We eat, you know, plant material, animal material, some of this, that contains microbes on it, and they're not really even residents. They could just be traveling through Maybe they're alive, maybe they're not. The DNA is probably there. DNA is pretty stable. And so we might end up, sequencing some of this material, and that doesn't really mean much. It could mean that, we all had wraps with a particular lettuce, and I'm not sure what microbes are on there. I know that sounds kind of gross, but maybe those microbes are like sending a signal through our gut, and it has no, real biological implications. Okay, so with that is the end. I have a bonus section, and I'm at 2:22, and my bonus section, I'll leave it to a vote, is I can talk about this recent paper. It's bonus because it has nothing to do with metagenomic annotation. It's about statistics, which we talked about some yesterday, and about differential abundance methods. So how about a vote of who wants to just get onto the lab, get your fingers going and do some lab work? Show of hands. Want to skip this? Who wants me to talk about this for the next 10 minutes? Oh, wow, it made me so happy. No, I don't care. I don't care either way. Okay, I'll talk. I made the slides, right? I had the slides made. Don't worry, I didn't stay up all night. Okay, so this is a paper that came out of my group. Jacob was the lead on it, but we all worked on this for many years. And so the idea here was to look at many differential abundance methods, right? And we're specifically focusing on methods that tend to just do two groups. Case control, whatever, something where you can put two groups. Okay, some of these methods will do more than two groups, but we focused on two groups. We also focused on 16S data in this, but many of these methods are applicable to metagenomic data as well. And we then wanted to compare basically the results across a whole bunch of samples, and a lot across different environments. And in all these particular samples we looked at, there was two groups, right? It could be IBD or not. It could be obese or not in humans. It could be salinity versus freshwater in oceans, or it could be, oh, what else do we have? Soil blueberry and something else. We had everything in here, which I'll show. And then we wanted to basically ask, well, how well do these tools agree on the results? And we knew they wouldn't, right? We knew they were going to be different, but we thought, okay, some tools are going to be really conservative and some aren't. So this was published if you want to check it out. And this is, I guess, one of their take-home plots, which is super busy. And so what this is showing is each of those different data sets on the x-axis, right? So we have human, marine, soil, lots of human, some freshwater, river stuff from Robyn's plastic stuff, or meta-analysis. And then what we have the other way, essentially, is our tools, right? And then the number here is just how many features they identified as significantly different between the two groups in those data sets, right? And so what you would hope is that across each data set, you would have a fairly consistent number. And we don't see that. So we see, oh, I hit the wrong one. We see that if we looked at this C. difficile one, that some of the tools are calling up to 3,000 ASVs differentially abundant. Others, like ADLX here, are calling 36 different ones. Huge difference in numbers. Now, we sort of expected that, but we thought that some tools would always predict more than others. And we sort of see that a bit. We see these tools overall, EdgeR, LeftC, LimaZoom, having quite a bit more, but not always, right? They're not always the highest one in these groups, which makes it kind of complicated. You do see, though, with things like LeftC, where some of these studies, you basically don't have any features called by the other methods, and we have huge numbers detected as different, right? So it's just characterizing that difference, making it very visible, I guess, to everyone, how much that does vary. And there's obviously tools like ANCOM and things that tend to be, I guess, more conservative. We also looked at then consistency between those tools. So just looking at overlapping features. And if we say a consistency score of 14 just means that those hits were shared across that many tools, right? And so what we see here is that for the majority of things, we basically don't have very many where they're consistently across. We get peaks here where LimaVoom, because there's two different versions, we get essentially a peak of 2, and then Wilcoxon with a CLR normalization is very low. So consistency is pretty bad across the board. And then the last thing I really want to mention about this, and this, I know this is really small, I'm sorry, but we also did a essentially sort of like a simulation to try to figure out a false positive rate. And for that simulation, what we did is we took the real data sets and we had two groups, remember? So we had essentially like a label, like C diff or not. And then we just randomly, we messed up those labels. We just assigned the labels equally to new samples within that same data. So you're hoping now that, you know, and we did this over and over and over, many repetitions. So you would expect not to find difference because we're just randomly assigning a label to it, right? So what this is showing then is then our number of significant hits. Most of them are on 0, which is good. But we did see for a few tools that we got significant number of hits. This is on a log scale for some of these data sets. And so LEFC, unfortunately, is here. EdgeR is here. And LimaVoom as well is here as well. So that's not great, right? You don't want to find random things based on random labels. And so we talk a lot more about that in the paper with some more details. I'm not going to go into it here. It's kind of depressing. You're like, this is depressing. It kind of is. I agree. Bit of a downer here. And then so the big question is, you know, what should you do, right? What tool should I pick then? And we don't have an answer for you, as always. We said it depends, like everything else. But in my lab, we have sort of started basically being pretty honest about a few different tools, running them, and actually just displaying those results. So I used Robyn's example, and we have a couple of others. This is from an environmental sample that Robyn's done, looking at different types of, I don't remember this now. This is water and microbes associated with biofilms with wood in the water. Yeah, it doesn't really matter. Anyway, it's cool stuff with groupings. And then, oh, did I cut off the labels? Oh no, right, sorry. Yes, I remember now. So then what we have is different taxa listed, and you can sort of see their presence in this heat map, right, across these groups. And now what we started doing is just running a few different tools. So in this one we used, did you use 3 different tools? You used ANCOM probably. There you go. ADLX. What was the other one? Maslin Rarefied. There you go. Okay. And then the way Robyn showed this is then she just did a heat map of essentially how many tools that showed that. So a darker red means all 3 tools found that significant between those 2 groups. And a lighter color just means that it was only found by one of those tools. And there's a few different ways you can do that, right? You can visualize this other ways, but we find it's pretty honest, pretty open to say, look, this looks a pretty good, this taxa is, you know, detected by 3 methods. Realistically, it looks really different. We do find more others that are different between our groups, but it's only detected by maybe 1 tool and not by others, so we're not as sure about it. Is it a statistically robust approach to, you know, clearly identifying significant features? No, statisticians would hate this approach, but it is a fairly honest way to present results, I think, to your audience who's reading the manuscript about what you think is a strong claim about what organisms are different or not. And so that's the best recommendation we have at this point until someone comes out with a better tool or some way to really get a better consensus across these tools. And that's where we are right now.